really all going to be about how to protect and preserve the value of your biological studies using the system. So before we start, a little bit about the Edge, for those that aren't aware of us before. The company's been around for a little over 10 years. It'll be 11 years in March. We're an independent company. We specialise really in the biological domain, so providing solutions for bio and pharmaceutical research companies. Our software is used by 4 of the top 10 pharmaceutical companies, and there, as well as a whole range of different middle tier and biotech companies. Our customers spread right from the west coast of the US in the west all the way through to Shanghai in the east, throughout Europe as well. What we do is we deliver solutions which for a range of different things. Today we're going to focus on the ELN aspects. We also provide solutions for assay requesting and planning, for data management and electronic lab notebooks, across disciplines such as DMPK, ADME, in vivo, screening, and biotherapeutics. We also have a compound management module, which is used for ordering and tracking of inventory work. If there's one thing we're passionate about, it's about helping you to manage your data and optimize your processes whilst keeping the value of your data. And that's going to be a key theme that we're going to be discussing today when we talk about electronic lab notebook features. So the objectives of today's webinar is really to provide you with information about how you can protect and preserve the value of your biological studies. We're going to review some of the challenges involved with that process. We're going to talk about our solutions there, and then I'm going to show you a demonstration so you can see the system in action. And then finally, we'll review the benefits and take any questions you've got at the end. So what are the challenges involved in providing an electronic lab notebook which can preserve the value of your biological study data? Well, here are 6 different elements which are raised as challenges. First one's around homogeneity, and we'll talk in more detail about that in a second. The second one's about the variability required, the number of different types of study. The third one is how to capture the structure of biological data as well as the pure information. And then we're going to talk a little bit about the challenges of managing workflow in the biology domain, where we've got multiple teams and individuals. And then we're going to talk a little bit about productivity and how that raises challenges, and finally the challenge of actually being able to exploit the data that you've captured whilst you've been using this electronic lab notebook. So the first one of those topics, homogeneity, this is really talking about the many different types of data that are associated with the data you record while you're doing a biological study. We're talking about things like metadata, that's information about information. You've got structured data, such as results, observations, and pure numeric observations, or maybe they're derived results from a number of different observations that you've taken. Then we've got, obviously, image data. We might be taking histology images or pictures of animals or other things that we're dealing with in a study. And then we also want to be able to capture unstructured information, such as our observations or conclusions or theory about this particular study and what we're observing during it. And of course, we also want to record signature data. So that means we want to record the fact that this data has been recorded on a specific date and it's been countersigned and witnessed by somebody else to make sure that there is a record of the intellectual property as it was recorded. So I've got a series of screenshots of taken from different laboratory notebooks, and we'll talk about those different types of data. So the first one is metadata. So in a laboratory notebook page, this would often perhaps be the title or the author or the data it was recorded on. So they're all pieces of metadata that you might be able to use to go and find the appropriate notebook, the appropriate person's notebook, maybe the appropriate experiment done on the date you're looking for. So they're all types of metadata for an experiment. Of course, we also want to be able to record structured information. So on this notebook page here, you can see there is a table of data, which is recording different facts, numeric, alphanumeric, and other types of data, structured, so it's recording the entity it's been measured against and then the number that's been recorded, and maybe matching a specific condition it's been recorded under. So this is a piece of what we call structured data. This is a relatively simple one. It's just a simple table with 4 columns and a number of different rows. But they can also be much more complex than that with multiple relationships within them. The next element is obviously capturing image data as well. Many biological studies involved capturing of images, such as gel images that you can see on this particular page here. But it might also be a histology image where we're looking at variation of cell structure during a biological study. And of course, we also want to record unstructured information. So on this page here, you can see there are some notes made by the user, some observations, and there's also, at the bottom of the page here, you can see there's signature information as well. So we've got some dates, and we've also got a witness and a counter signature on the page to prove that it was done when you said it was done, and the data has been checked. The next area is around variability. So obviously there are multiple different types of study that are in operation within an organization, specifically those doing pharmaceutical or biopharmaceutical research. And those can vary in type from what we call routine studies, so which might be something like a routine screening assay, or it might be a routine study such as measuring the metabolism of a particular drug. They run to a strict protocol, they're always run the same way, and they need to adhere to the protocol that's given. However, at the other extreme, we might have a non-routine study which is completely exploratory. So we might have really not know all the experimentation that's going to go on during the study when we start it. So we might run some initial tasks during the experiment. Then according to the results we observe, we might define new tasks that we're going to do. And so it's a much more less routine and much more exploratory study. And we need to be able to cope with the full continuum between these completely defined studies with a strict protocol all the way through to the more non-routine exploratory studies. The other area is being able to capture the structure of the biology that we're evaluating. So biological data, it has dimensionality, and that dimensionality really matters. It matters because unlike perhaps some other disciplines where you might have sort of fairly simple data types which have fairly simple relationships, in biology we're dealing with more complex organisms or organs within an organism, where we're taking lots of different observations and exploring the biological space that we're evaluating during our study. So that means we need to capture lots of different conditions, observables, and other settings of the assay, and then hold that data together. Because in biology, the value of an individual number is really only when it's associated to all the associated conditions that describe that number. It's no good just saying, you know, the result is 25 if we don't know all of the conditions that have been used in order to measure that result during the study. And of course, we also might have lots of data too, and be able to visualize it in multiple different ways and compare one observable to another, or trend data over time, or look at things like kinetics data. So the structure of the information is really important that we keep that. Also, we need to think about capturing that structured data and also how we're going to use it afterwards. So like there's capturing data at the time of capture. There's also reviewing that data maybe later, so we're doing some quality control on it. And then also we've got to think about how we exploit that structured data afterwards, because it's not just about a single study. We also might want to look at a whole organism and think about, you know, how it varies over lots of different studies that we're using. So if we can make our life easy by just recording the information now, but you can find it leads to a lot of headaches later. So for example, if you just record the data as text and put it into your notebook and record it, that's all very well. But if later you need to be able to trend that data or look at statistical variation of it, then having it, you know, in a textual document is not very useful. So we want to be able to capture the structure of the information so we can maximize its value. The other element is workflow. So biology is very much a team-based exercise. You've got multiple teams working together for the overall goal of completing this study. So it's team-based, multiple teams working together. The study data itself can be collected by multiple different users and teams. So we're essentially creating one study that's made up of multiple activities that are distributed across different teams that are all recording different information that we then want to be able to bring together in order to make our conclusions. It also has to deal with both internally generated data and also potentially externally generated data as well. So for example, we might outsource the animal work to a contract research organization, but bring in the samples that are generated, do their analysis, and make our conclusions afterwards. So we need to be able to track that workflow. Here's a typical example for here, this workflow here. So we're going to order our animals, we're going to order our compound, we need to formulate the compounds, that will be done by a formulations group. We need to cage and group the animals together and condition them ready for the study. That will be done perhaps by an animal welfare group. And then we're going to treat the animals with the formulated drug and then sample them. And then, of course, we need to sample those, analyze those samples perhaps in a centralized bioanalysis group. So really, if we think about this networked organization with lots of different departments working together and maybe external ones as well, really the idea of a personalized notebook, it really doesn't make a lot of sense in terms of the data itself. The next challenge is really about getting people to adopt the software, and one of the challenges of that is to help them be more productive. And there's generally two types of productivity. There's the personal productivity, which essentially says, how does this notebook help me to be more productive? I don't care about the company, it's me that matters because I'm the one doing the work. So if you're going to make me do things which slow me down, then I'm not going to be interested in using it. The second area is about corporate productivity. So this is basically, it's not about an individual. It could be a little bit about a team, but it's really about the overall program that's trying to move forward. So we want to be able to make sure that things we do don't affect other people's productivity in the organization. So we can work together to make sure that the whole project works efficiently. Now, it's really important that we take this seriously, because for an individual, if you don't give them a reason to use the system, then even if there are benefits to the corporation from recording data in the right way, they're not going to use it. So, and typically we might hear something like this: I've got too much work to do to look at software, or something like, you know, Excel works, what's wrong with that? Just leave me to it. If we do this, basically, the trouble with things like Excel and Word and file shares is the data might be fine for the individual, but there is no, you don't get the value of where you can work as a team, and the corporation certainly doesn't get the value of the data that's being generated. So it's important that we listen to these messages and we give them benefits. So, you know, there's a number of different things that we do will help someone to understand that actually this is going to be a time-saving device, and enable them to look at their data much more clearly and with less manual effort. And don't get me started on Excel. That could be a whole webinar. The last thing then is the challenge of how to exploit the data afterwards. So we need to be able to capture this data so that we can use it for things like quality control, both within an individual study and also across time as well. So we're looking at trends from data. We might want to do cross-study analysis, and also to be able to understand sort of complex disease models, which are bringing data together from multiple studies that are being run. So there's a number of challenges. So how are we addressing this? So what we need is a system that can combine demand tracking, so that's the, if you like, the demand for studies to be run, with an element of workflow support, so it can help teams work together to generate a study and to execute it. We need it to manage structured data, so that the data we're capturing during the study is available, stored with its structure, so it can be exploited afterwards. We also want a place where we can write up as well. So the thing with a lot of biological studies are, it is a complex topic and it does need interpretation. So often there's a reluctance to store the structured data because of a fear that somehow the results, if they're independent of their interpretation, can be a dangerous thing, you know, in an organization. So what we need to do is provide an environment where we can capture the key results, the structured data, and also allow them to capture their interpretation of that data so that people can view the results in its full context. And of course, we want to make sure that we can manage the communication between different people during that workflow as well. So we've created a system. It's been designed by biologists for biologists, and you'll notice there are a number of elements to it which helps biologists to work more efficiently. So the first one is the system needs to handle this study dimensionality. So we have this feature called elastic templates, which basically mean that a single experiment can stretch to meet all of your data. So it understands about things like treatment groups and subgroups and relationships between data. It also allows you to include your analysis within the same notebook page. So you've got consistency, so we're always analyzing data in the same way, and therefore can compare data and trend data across time. It also understands about your scientific data types, so it can read the data files that are generated from your instruments or from observations that you've captured during the purpose of the study. As I mentioned, it's got the built-in analytics, so it can do things like statistics for looking for variation over a study. It can also visualize your data in terms of graphs and charts, and also do things like curve fitting and non-compartmental and other types of analysis during the study itself. And they're all elements that talk to the productivity. So what we're trying to do is provide a single continuous environment which the biologist can use to run the whole study, rather than having to copy, paste and transform data, so it can be analyzed with other pieces of software. It's all instruction driven, so that means they can simply execute a protocol and click through the steps, so it guides them through how to use it. That lowers the learning barrier. I mean, it's much more easy to use in the system, much easier. So how does it work? So what BioRails does is it provides a system for managing the protocol and also the workflow. Everything in the system is defined as a folder which can contain version content. That content can be presented as both an electronic laboratory notebook page, where there are various sections for the data being captured. So that's the metadata, the structured data, the image data, and also the unstructured information. And it also allows you to present that same data as more of a workflow view, so you can see it in terms of a breakdown of tasks that need to be done that can be distributed amongst different people. The folders can also be printed directly to PDF for archiving purposes, and that will contain all of the information, including, of course, the structured and unstructured information in the PDF. And the signature information so it can be used as a defensible document in court. It allows you to add files, including Office documents, PDFs, and these Morphit templates, which are used for data analysis, as well as things like image data, which can be annotated. So what you're doing is storing your unstructured data and your structured data together with the content, so that it's searchable by the dimensions, and also you can evaluate the interpretation of that data as well. Of course, it's an electronic lab notebook, so we also want to protect the intellectual property that's being captured during this exercise. So the system provides electronic signatures. So there's a signature record. It's a permanent record in the system that protects the content. It's then therefore tamper-proof because it has something called a signature hash, which is a digital fingerprint for that content, such that if any of the content has been tampered with at the back end, then that signature is invalidated, and you know the data is no longer safe. And that provides a defensible document that can be taken to court and cross-match with the system. The system provides a signature workflow so that you can author and witness content and manage that overall workflow through communication through the platform. So via both email communications requesting either a witness signature or whatever, and also there are notifications in the application itself. The signature process can be manual, so you'll say, I'm signing this now. It can also be event-driven, so as you take an experiment through its natural cycle, so if you like, from initiation through planning, through various stages, eventually it will get to something called a signature stage, where it will ask you for electronic signature and then a witness event before it moves on to any further stages. So what we're going to do now is I'm going to show you the system, and I'm going to show you an example of how to create an experiment in the system. We're going to capture some structured and unstructured data. I'm going to show you how to work as a team, so we can pass around different aspects of a workflow to different team members. And then I'll show you quickly how we can exploit the data afterwards, and afterwards we'll take any questions. Okay. So this is BioRails. It's a web-based application, and I'm just going to log in. So I'm going to log in as someone called Barbara Bryson. And Barbara here she's a laboratory manager, so it could be a study director. And what we're going to do is just go ahead and create an experiment. So we come to the menu here. I'm going to create a new experiment. And experiments can, if we have access to multiple teams, then we can store the experiments under whichever team that we're operating under. So this one's going to be, I've set this demonstration system up to be set up for a team working inside a contract research organization. But it could equally be a pharmacology team inside of a pharmaceutical company, or any other type of biologists working in their appropriate team. And then we've got, you'll see it generates a study identifier, which can be configured. And basically here, I can pick the types of study that I want to run. So we'll start off by running what's called a generic experiment. So all this is going to do, it's going to ask me which project, what the objective of the project is. It's going to ask me to pick a generalized assay type, and then I can specify which compound this study is going to be operating on. All of these forms are completely configurable, so I could have made it about a target or an animal or any other subject, or had a completely different form on here, which is completely designed by you when you set the system up. You can see I can also pass this experiment to one of my colleagues if I want to. I can also give them some basic scheduling information in here. So we're going to start it today, and it's going to be finished in 2 weeks' time. We'll just confirm that, and it will create my study page, which is essentially a more or less blank electronic lab notebook page. So if we edit the page now, you can see I can pick which project it's for. So I have one on my list, that's the global project. I can set an objective. So I'm going to evaluate a set of histology images in here, this particular study. I can also look up the assay type, and this is a hierarchical dictionary that I've entered into the system to specify what sort of study I'm running. So we'll just say it's a toxicology study, and I'm going to be dosing a particular compound. So just for a moment about these fields, all of these fields are built up inside the data catalog of BioRails, where all of the master data is for the study. So that includes, here I'm looking at compounds from my chemistry registration system. These assay types have been built up inside the BioRails catalog. And then also the objectives is free text, and you can see the project is made up of my registered projects for this particular notebook. Okay, so that's the basics of this. I can now indicate that this study is started by moving it to active. So we're now in an active state. So really all I've done is specify the high-level information for the study. And now what I can do is build up my study by adding information. So I can add Excel workbooks if I want. I can add files. I can add subfolders to the experiment. I could add notes and observations. So maybe we'll set in the first set of, let's call it the theory section. And here, of course, it's free text, so evaluate these histology images according to standard protocols. Not being able to type that. And we'll save that, and that gets saved as text. Again, you can copy and paste from Word and other things. So we now have a theory section. Then we might want to add some files to it. So what I'm going to do is I've got some here, some histology images that have been recorded, and I can just drag and drop those on the page. So let's do a few in here, and then upload them to the system. So now on the page you can see I've got my structured data at the top, I've got some unstructured theory here, and underneath I've got various images that have been recorded. And I might want to annotate these images, so I'll just go on there, add an annotation, and you select the area you want to annotate. So do that, and we'll say this is a cell or it might be, I think these are actually tumor cells inside the, so we'll add a different one. So this is non-uniform tumor. You'll notice that the annotations are under here, and if I highlight the annotations, you can see they're highlighted on the image above. So we'll close that. And again, I could go through and look at the others. And then maybe I want to add some other files. It could be some raw data files that we're adding. I could also add some conclusions if I want. So we'll add some conclusions. So we'll make that our conclusion. So now what I've got is a very simple experiment that's been written up with various different sections in it. And so I might want to sign this. So I haven't finished the experiment yet, but I'm going to sign the intermediate version. So again, I just go in here. This user doesn't have rights to sign, so let me just log in as someone who does. I might have to go in this button. Refresh the page here, and I'm going to sign this page. So here you can say, type in your username and a comment. And then I can select a witness. So I'll send that to Arthur Crow for a witness. And then I can sign that version. What that does then is to sign this current version. So it's applying an electronic signature. You can see details of the signature here. So I've got one version of this particular experiment. I can also see the PDF if I want from here, or I can go back to my original experiment. And you can see now it says signed by Fox Mulder, that's the user I'm logged in as, on the date and time here. If I then log out of here and log in as Arthur Crow, well, first of all, Arthur will get an email. Also, if he looks on his list of signatures, you can see now there's a request come through for signature of this particular experiment. So we've got details of the content of the experiment. If I want to actually have a look at it, I can just come in here. You can see I can review it. If I like what I see, I can countersign it, or if I want to, I could reject it with a reason, and that would be rejected and sent back. So we'll countersign this one. Actually, that's not. And then obviously I can witness it then. And that applies the second signatures to this particular experiment. Now I can, if I want to print an archive document which has the recording of the intellectual property, I can just print the print to PDF, and we'll get out a high quality PDF print of the study report. Okay, this one's obviously quite simple, but you've got the experiment title, the author, the version number. At the bottom of each page, we've got the signature hash and also the details of its reference into BioRails as well. And then I've actually got the data. All the images are in here as well as the structured data that was recorded. And of course, at the end, I've got details of electronic signature, so we can use that in our defense in court. So this version of this experiment has now been signed and witnessed. I could also trigger the same workflow. It doesn't mean that I can't change the experiment now. What it means is the version of that experiment as now signed and witnessed is now fixed in the software and can't be modified. If I want to, I can create new versions simply by editing this, making changes, and all those changes will go into the audit history for the experiment. The other way of triggering an experiment is to move it from active to the signed state. When I move it to the signed state, it's going to automatically ask me for a signature in order to move further to the published state. Okay, so that was a fairly simple example. Now I'm going to show you a more complex one. So I'm going to log back in as Barbara Bryson. And this time I'm going to go into this microsome assay queue here, so I can actually see which studies have been demanded by other people in the organisation. So here you can see that Walter Skinner from the chemistry department has asked me for some results for these two compounds here. So I'm going to put them in the cart. This time I'm going to create an experiment by a slightly different route. So it's the same experiment creation, except this time I'm going to pick up the protocol details that were requested by that chemist when he asked for the study to be run. The other thing I can do here on this example is I might want to run this at a contract research organisation instead of doing it internally. If I want to do that, I can still create the experiment, except this time instead of tracking all of the data that's generated internally, it's going to be used to have the results that come back from running it at the contract research organisation. In this example, I'm going to run it internally, so I'm going to use this example here. So we're going to run our microsome stability results workflow protocol for this experimental page. And again, I could distribute it across the team members. I can also put scheduling information in. So we'll save that. And this is deploying now a different sort of laboratory notebook page, which combines the element of structured data capture, unstructured information observations, and also the data analytics as well. So here you can see I've got a table above. It knows which compounds are going to be tested. It's also got a space then to record the results. So I've got some graphical results above and also some numeric results. So here we're measuring these compounds against the microsome. It's mice microsome, a particular concentration of compound, and we're going to measure the intrinsic clearance, scale clearance, and other factors. And we do that using an embedded template, which you can see here. So what I'm going to do, I'm just going to verify one thing before we start. All we're going to do is run that template, which will then bring data in from structured data sources, scientific data files, and then it will allow me to interpret the data. So I'm just going to okay, so all I have to do now is just click the link here. I'm going to click the link, and that will open up the biological template for managing the data analysis within this particular microsome study. So it's opening on the other window. I'll bring it over as soon as it's ready. There we are. So this is my template. Inside here we've got our workflow down the left-hand side that's going to help us to run through and capture the structured data. So we'll start off with the settings page. You can see it understands which experiment it's done from. I've got the basis of the experimental details here. I'm then going to import a mass spec file, which is going to come from here. So we're going to bring in this example here, which is basically a mass spec file coming from a mass spec instrument. So it's got all the peak areas and other data in it. So now, normally we may have to have opened up a different piece of software to import that mass spec file and do the calculations in it. But in this example here, I can bring it in and interpret it within the actual BioRails notebook itself. So we've brought the data in. You can see some of the data has been knocked out because the areas are below the minimum threshold. I can review the batches that have been tested. If there is any calibration data, I can review the calibration curves. There isn't in this example. And then move into my analysis. So here what we got for every batch that's tested, you can see I've got a plot of the log of the % remaining over time, and also I've got my true controls, so I can tell whether things are behaving or not. I've plotted both a linear plot of the log and also an exponential plot of the % remaining. On the right-hand side here, we then derive from that data a set of results. So I've got a K value with a standard error, the half-life for the compound as it's metabolized by the microsome, and also the area under the curve to infinity, which is a measure of the total amount of drug that's been metabolized. These results can also be interpreted and viewed for every compound in the set here. So you can see these will run against, these are rat microsomes. And I've got my clearance values on the right-hand side. Also, it scaled the clearance according to for humans, so modeled from the rat microsome to the predicted human clearance. And finally, I've got my results here categorized as high, medium, and low. So the last step now is to post that data. And what that does is it captures that structured information that we've taken from our mass spec, and it's brought it over into the system. So what we'll find is, just refresh the page. You can see now it's now combining graphical results for the data that's been generated in this particular run, and it also has captured all the numeric data, that's structured data that's searchable and usable in the system. And I also have my graphs, so again they're all annotatable. So if I want to, I can annotate one of the columns to show that it's good or bad. I can also add my interpretation, so I could add a conclusion section. I can add any raw data files as well. And of course the final thing I'll do is to sign the data and then publish the data afterwards. So I won't go through that because I've already shown you that. But what I want to show you here is the fact that we've combined the structured data also with any observables and assets like files and content. They're all managed in the same system. Once that data is captured, we can then start searching it. So here I've got an example of a query which is searching across all of the studies for microsomes. So it's doing cross-study analysis. And here it's showing me the trending in the standards data across all those studies. And in this filter, you're seeing I've got the individual results. That's really important because it's going to allow me to actually manage the quality control of my data. And I'll open up a quick other binder which will show you an example of how we can exploit that data. So in this example here, what we need to do is do the quality control of our microsome study. And I need to be able to compare the performance of the standards in the current study in the context of all the previous studies that have been run for the last 6 months. What this binder has done, as I've opened it, it's queried the last 6 months worth of data, and then it's plotted the clearance of the control data, the standards, sorry, in this particular study, and plotted them as stars on the chart here. And then all the blue points are the same standards clearances measured over all the studies run over the last 6 months. So this immediately gives you a visual cue that these results are within 2 standard deviations of the mean of the clearance for these standards in this assay. And if it falls outside this range, I can then say, well, there's something gone wrong with the protocol or the way I've executed it, and I can withdraw that study because it fails quality control. And we can only do that because we've captured structured data together with the unstructured observations. And that's what makes this system very powerful, because you're capturing the structured data with all its dimensionality and the unstructured data together with the IP protection. Okay, the last example I want to show you is a more complex study where we have multiple steps. So I'll create a new experiment. So I want to show you the workflow aspects of this one. So we'll just come through here. I think it's, let's look at bioanalysis. We'll do this one as an example. So here what I'm going to do is build up a study and add multiple tasks into the same study. So this is just a bioanalysis study. I'm measuring a tissue sample. I'm going to record which study it came from, its batch, route of administration, dose, etc., and then the measured tissue concentration. What I can do now, I could add many instances of this within the same study. So first of all, I'm measuring the first sample. I'm then going to add a second sample using the same protocol. I'll then add a third, and then I'm going to show you how to distribute those across different people in different teams. So add another one. Now, if I go up to the primary study, and we view the contents view, or I can actually go and change view of this to dashboard view. What we have here is we've got the main study details above, and underneath I've got a subtask here. So I might want to move this subtask out to somebody different. So again, I could pass it across. So normally it would be to a bioanalysis team, for example. That allows me then to distribute the work across different teams. And then if that person logs in, they would see on their input list that a task has been assigned to them. They can also subscribe to get an email to say there is a new bioanalysis task to be run to you. And these workflows can be made up of multiple steps that can be distributed between different departments. Okay, that concludes the demonstration part of this. Just return to the slides. So what I've shown you basically is a system which allows you to capture structured information, unstructured information, either from a defined routine protocol, such as that microsome study, or for a more exploratory study, such as the tissue analysis one I just showed you, where I can build the study up with different steps as I go. I also showed you how you could distribute that work across multiple users in a team environment. I've also shown you how you can protect the intellectual property generated, so you can capture your unstructured notes, you can build up your study report with its interpretation of the data and results, you can export that to PDF and archive it, you can also share those results out with other people in the organization. All of those results are stored and searchable. You can search both the unstructured data in a sort of Google-like search. You can also search the structured data using the reports, as I showed you earlier on, where I was looking at controls and standards over time. So all of this means that the intellectual property that's generated is protected, and that's built into the daily workflow of the scientists. It means the data captured in experiments can also be analyzed, whether it's for quality control purposes, or whether it's for long-term trending and analysis of data. All the data can be analyzed in order to create secondary knowledge, which again can be written up. This also means that I can run multiple studies and create new studies which are simply derived to evaluate the results generated from other studies. So we can do things like cross-study analysis, trending of data. We can also visualize and do statistics on that data as well. And that's all because we've captured data with the dimensions of the studies. So it means all our biological research is managed in a single system, but the intellectual property and also the data itself, and it's all workflow enabled, so you can use it to collaborate between different teams, also between different organizations, so out to contract research organizations and back, for both routine data and also more exploratory work. This particular notebook that you've seen today is in use at lots of different companies. A couple of them I can talk about. One is Evotec, where it's used for their DMPK services that use BioRails as both their electronic lab notebook and also their data management system. So it means the DMPK scientists only need to use one system. They can use it to track their customer requests for DMPK studies. They can use it to plan out the studies, distribute the work amongst their team, and also then record the structured data and their observations and interpretation of the data before it's released back to their clients. Another example customer is Lundbeck Pharmaceuticals, where they use it as their electronic lab notebook for all of their biologists that are involved in things like behavioral studies, in vivo studies, ADME, and DMPK as well. There's about 400 scientists using the system there. What this means basically is we're giving you a system which can be used to support all of your biologists, so it's a better long-term investment for supporting all of your different studies.